Welcome, gentle listeners, to a journey into the invisible realm that shapes our entire world. Before we begin this contemplative exploration, please consider subscribing to our channel for more sleep-focused, educational content that nurtures both rest and wonder. In every grain of soil, in every drop of ocean water, in every breath we take, invisible architects work tirelessly, building and rebuilding the world we know. Close your eyes, dear listener, and imagine for a moment that you could shrink yourself down to a size so small that you could walk among the true architects of our world. Not the engineers who design our cities, not the forces of wind and water that carve our landscapes, but the microscopic beings that have been quietly, patiently, lovingly crafting the very foundation of life on Earth for billions of years. These are the microbes, bacteria, archaea, fungi, countless other microscopic organisms that exist in numbers so vast they defy human comprehension. If we were to count every microbial cell on our planet, the number would exceed the stars in the observable universe by factors of millions. Yet they work in silence, their contributions invisible to our naked eyes, their impact immeasurable, yet largely unnoticed by the conscious world above. Tonight, as you drift towards sleep, we shall travel together through their hidden cities, explore their ancient neighborhoods, and witness their magnificent constructions. We shall see how these unseen architects have shaped not just the landscapes we know, but the very air we breathe, the soil that feeds us, and the bodies we inhabit. Picture, if you will, a world without these tiny builders. The earth would be a barren, lifeless rock floating in the cosmic darkness. No soil would exist to cradle the roots of plants. No oxygen would fill our atmosphere. No cycles of nutrients would flow through ecosystems. The very concept of life as we know it would be impossible without these microscopic artisans who work around the clock, through every season, across every continent and ocean. But our world is far from barren. It pulses with life, it breathes with complexity, it flows with interconnected systems of breathtaking beauty, all because these invisible architects never rest from their work. They are the foundation upon which all visible life is built, the hidden infrastructure that supports every forest, every coral reef, every mountain meadow you have ever admired. Let us begin our journey at the very beginning, in the deep time. When our planet was young and these microscopic architects first began their work, picture the early Earth, some 4 billion years ago, when the surface was still cooling from its violent birth, in the primordial oceans, warmed by underwater volcanic vents. The first microbial contractors set up their workshops. These ancient bacteria were unlike anything we see today, yet they carried within their tiny cells the blueprints for everything that would follow. They were the first to harness energy from their environment, the first to build complex molecules, the first to reproduce and pass on their architectural knowledge to future generations. Slowly, patiently, over millions of years, these pioneering microbes began to modify their environment. Some learned to capture sunlight and use its energy to build organic compounds, releasing oxygen as a byproduct of their cellular construction work. This oxygen, initially toxic to most life forms, gradually accumulated in the atmosphere, creating the oxidizing environment that would later support more complex forms of life. The Great Oxygenation Event, scientists call it, was perhaps the most dramatic renovation project in Earth's history. These microscopic architects literally redesigned the atmosphere, transforming a world of methane and carbon dioxide into one where oxygen-breathing organisms could eventually thrive. It was construction work on a planetary scale, carried out by beings so small that millions could fit on the head of a pin as eons passed. These bacterial architects diversified their skills. Some became specialists in breaking down rock minerals, slowly transforming solid stone into the chemical building blocks of soil. Others mastered the art of nitrogen fixation, taking the inert gas from the atmosphere and converting it into compounds that plants could use for growth. Still others became the recyclers of the biosphere, breaking down dead organic matter and returning its components to the cycles of life. Each microbial lineage developed its own architectural specialty, its own construction techniques, its own role in the grand project of building a living planet. And unlike human architects, who design buildings to last for decades or centuries, These microscopic builders created structures and systems designed to function for geological ages. Now, let us descend into the earth beneath our feet, into the dark, rich world of soil, where some of nature's most skilled architects ply their trade. If you have ever wondered at the fertility of a garden, the productivity of a farm field, or the lush growth of a forest, you are witnessing the handiwork of billions of microscopic soil builders who labor in the darkness below. Soil, that humble, brown substance that we so often take for granted, is not simply ground-up rock. It is a living system, an underground metropolis more complex and bustling than any human city. In a single teaspoon of healthy soil, there are more microbial inhabitants than there are people on Earth. Each of these tiny residents has a role to play in the vast construction project that transforms dead rock into the living foundation of terrestrial life. Picture this underground world as a vast network of microscopic neighborhoods, each with its own character and function. In the spaces between soil particles, bacteria move like tiny bulldozers, breaking down organic matter and mineral fragments. They secrete enzymes, molecular tools that can dismantle the most complex organic compounds and reduce them to their component parts. These bacterial contractors work in teams, each species specialized for particular demolition and construction tasks. Some excel at breaking down cellulose, the tough structural material of plant cell walls. Others specialize in decomposing proteins, lipids, or other organic compounds. Working together, they can completely recycle the remains of fallen leaves, dead roots, and other organic debris, transforming death into the raw materials for new life. Among the most remarkable architects of the soil realm are the fungi, whose thread-like structures, called hyphae, create vast underground networks that dwarf human transportation systems in their scope and efficiency. These fungal threads, finer than spider silk, yet stronger than steel cables on a microscopic scale, weave through the soil like an underground internet, connecting plants, sharing nutrients, and facilitating communication across the forest floor. Imagine these fungal networks as the highways and bridges of the underground world. A single fungal individual can extend its hyphal network across acres of forest floor, connecting hundreds of trees and thousands of smaller plants in a web of mutual support and resource sharing. Through these living conduits, nutrients flow from areas of abundance to areas of need. Water moves to drought-stressed plants, and chemical signals carry information about threats and opportunities throughout the forest community. The fungi are master builders of soil structure as well as soil networks. Hyphae wrap around soil particles like microscopic rope, binding loose sediments into stable aggregates. These fungal construction crews create the pore spaces that allow air and water to penetrate deep into the soil. The channels that plant roots follow as they explore their underground world, and the stable architecture that prevents erosion and maintains soil health. Some fungi form intimate partnerships with plant roots, creating structures called mycorrhizae, literally fungus roots. In these collaborative constructions, fungal hyphae extend the reach of plant root systems by hundreds of times, exploring vast volumes of soil that the roots themselves could never access. In return for this service, the plants provide the fungi with sugars and other organic compounds produced through photosynthesis. This ancient partnership between plants and fungi is one of the most successful architectural collaborations in the history of life. It enabled plants to colonize dry land, allowed forests to spread across continents, and continues to support the productivity of nearly every terrestrial ecosystem on Earth. Without these fungal contractors, our world would look very different. Indeed, likely a landscape of sparse, struggling plants rather than the lush forests and grasslands we know today. Among the most essential of soil microbes are the nitrogen-fixing bacteria, microscopic alchemists who perform one of the most important chemical transformations on our planet. These specialized architects capture nitrogen gas from the atmosphere, that inert, unreactive gas that makes up 78% of the air we breathe, and convert it into forms that living organisms can use to build proteins, DNA, and other essential Molecules, the process of nitrogen fixation requires enormous amounts of energy and incredibly sophisticated molecular machinery. The key enzyme, called nitrogenase, is a biological marvel that can break the triple bond holding two nitrogen atoms. Together, one of the strongest chemical bonds in nature. These bacterial architects must protect this delicate enzyme from oxygen, which would destroy it, while simultaneously providing it with the energy and electrons it needs to perform its transformative work. Some nitrogen-fixing bacteria work as independent contractors, living freely in the soil and sharing their nitrogen products with the surrounding community. Others have entered into exclusive partnerships with specific plants, taking up residence in specialized structures called root nodules. In these cozy bacterial apartments, built by the plant but furnished by the bacteria, the nitrogen fixers work around the clock, providing their plant partners with all the nitrogen they need for growth and reproduction. The relationship between legume plants and their nitrogen-fixing bacterial partners represents one of agriculture's oldest success stories. Long before humans understood the chemistry involved, farmers discovered that planting beans, peas, clover, and other legumes could restore fertility to exhausted soils. They were unknowingly employing microscopic architects to rebuild the nitrogen infrastructure of their agricultural systems. In natural ecosystems, nitrogen-fixing bacteria serve as the primary architects of productivity. They determine how much plant growth an ecosystem can support, how much carbon can be stored in vegetation, and how much food is available for the entire web of life that depends on plant productivity. Without these microscopic nitrogen architects, our world would be a much less green and less productive place. As we drift deeper into our contemplative journey, let us now turn our attention to the vast blue realm that covers more than 2/3 of our planet's surface. Here, in the endless expanse of the world's oceans, microscopic architects work on an even grander scale, shaping not only marine ecosystems but influencing the climate and chemistry of the entire Earth. The ocean, to our human perspective, appears as a relatively uniform blue wilderness, seemingly empty of the visible complexity we observe on land. But this perception could not be further from the truth. The ocean is filled with invisible cities, microscopic metropolises that dwarf human settlements in their scope, complexity, and influence on the world around them. In every drop of seawater, there are millions of microbial architects at work. Bacteria, archaea, tiny algae called phytoplankton, and countless other microscopic organisms labor continuously to maintain the ocean's chemical balance, process nutrients, produce oxygen, and support the entire marine food web. Their construction projects span the entire water column, from the sunlit surface waters to the deepest ocean trenches. At the ocean's surface, where sunlight penetrates the blue depths, microscopic photosynthetic architects work tirelessly to capture solar energy and convert it into the organic compounds that fuel marine life. These phytoplankton, tiny floating algae and bacteria, are perhaps the most productive builders on our planet. Despite their microscopic size, this surface dwelling architects produce more than half of the oxygen in our atmosphere. Every second breath you take was created by these tiny marine builders as they go about their daily work of photosynthesis. They are the hidden lungs of our planet, an invisible source of the life-giving gas that sustains all air-breathing organisms. Picture these phytoplankton as microscopic solar panels, each one equipped with sophisticated light-harvesting machinery that can capture photons from the sun and convert their energy into chemical bonds. Some species have evolved beautiful geometric shells of calcium carbonate or silica, intricate, microscopic sculptures that serve both as protection and as optical devices too. Focus and distribute light within their cells, the timing and location. A phytoplankton bloom's massive population explosions of these microscopic architects shape seasonal patterns across the globe. In spring, when warming waters and increasing daylight trigger massive blooms, the ocean changes color as billions of tiny builders multiply in the surface waters. These blooms are visible from space, vast green and brown swirls that can span hundreds of miles, representing construction projects of almost unimaginable scale. As these surface architects build organic matter through photosynthesis, they form the foundation of the marine food web. Tiny zooplankton graze on the phytoplankton like microscopic cattle. Small fish feed on the zooplankton, larger fish eat the smaller ones, and so on up the food chain to the great whales and other apex predators of the sea.
Acris.
Primary contractors. The ocean would be a biological desert, but the work of oceanic microbial architects extends far beyond the sunlit surface waters. In the vast darkness of the deep ocean, where no sunlight penetrates and temperatures hover just above freezing, specialized bacteria and archaea have constructed some of the most remarkable ecosystems on Earth around underwater volcanic vents, where superheated water rich in minerals erupts from the ocean floor. Chemosynthetic bacteria serve as the primary architects of life. These remarkable microbes have learned to harness the chemical energy released by reactions between volcanic minerals and seawater, using this energy to build organic compounds in much the way that surface phytoplankton use sunlight. These deep-sea bacterial architects create oases of life in the ocean's deepest places. Their chemical construction work supports entire ecosystems of specialized organisms, giant tube worms, ghostly white crabs, and bizarre fish that exist nowhere else on Earth. These deep-sea communities represent alternative blueprints for life ecosystems built on chemical rather than solar energy, demonstrating the versatility and creativity of microbial architects in the sediments of the deep ocean floor. Other microscopic builders work at a more leisurely pace, slowly processing the rain of organic matter that drifts down from the surface waters. These benthic architects, living in sediments that may have been accumulating for millions of years, work in geological time, slowly recycling nutrients and maintaining the chemical balance of the deep ocean. Some of these deep-sea microbial architects have been living and working in the same sediment layers for thousands of years. Their metabolism slowed to an almost unimaginable crawl. They represent perhaps the most patient constructors on our planet, building and maintaining their microscopic communities across timescales that dwarf human civilization. Among the most important work performed by oceanic microbial architects is their management of the global carbon cycle. The ocean serves as the planet's largest carbon reservoir, and microbes are the primary controllers of how carbon moves between the atmosphere, surface waters, and deep ocean storage. When marine phytoplankton photosynthesize, they remove carbon dioxide from both the atmosphere and surface waters, incorporating this carbon into their microscopic bodies. Some of this carbon is quickly recycled as the phytoplankton are eaten, and their remains decomposed by bacterial architects in the surface waters. But a significant portion sinks into the deep ocean, where it may remain sequestered for centuries or millennia. This biological carbon pump, driven entirely by microscopic architects, helps regulate Earth's climate by controlling the amount of carbon dioxide in the atmosphere. During ice ages, changes in ocean circulation and microbial activity can draw down atmospheric carbon dioxide levels, contributing to global cooling. During warmer periods, increased microbial respiration can release stored carbon back to the atmosphere. The ocean's microbial architects thus serve as planetary climate engineers, their collective activities helping to maintain the delicate balance of atmospheric gases that makes Earth habitable. They are the thermostats of our planet, working continuously to buffer changes in the climate system and maintain conditions suitable for life as our journey continues through the realm of unseen. Architects, let us now lift our perspective skyward into the vast ocean of air that surrounds our planet. Here, in the seemingly empty space above our heads, microscopic builders work on construction projects that span the entire globe, shaping weather patterns, influencing climate, and even affecting the formation of clouds and precipitation. Atmosphere, like the ocean, appears deceptively uniform to our human senses. We perceive it as empty space, a transparent medium through which light travels and birds fly. But this aerial realm is actually filled with microscopic architects, whose activities influence every breath we take and every weather pattern we experience. High above the Earth's surface, bacteria and other microorganisms drift on air currents, like aerial plankton, traveling vast distances and seeding new environments with their genetic blueprints. These airborne architects face extreme challenges: intense ultraviolet radiation, desiccation, extreme temperature fluctuations, and the constant stress of wind and turbulence. Yet they have adapted to these harsh conditions and learned to use the atmosphere as their construction site. Perhaps the most remarkable atmospheric architects are those microbes that serve as cloud condensation, nuclei, the tiny particles around which water droplets form in the sky. Without these microscopic builders, clouds, as we know them, could not exist, and the entire water cycle would function very differently. Bacteria, fungal spores, and other organic particles released from the Earth's surface rise into the atmosphere on convection currents and thermal updrafts. Once aloft, these microscopic architects provide the nucleation sites that allow invisible water vapor to condense into visible droplets. Each cloud droplet forms around one of these tiny builders, making every cloud a massive aerial construction project involving billions of microbial architects. Some bacteria have evolved specifically to enhance their effectiveness as cloud condensation nuclei. They produce special proteins on their surfaces that encourage ice crystal formation, making them particularly effective at triggering precipitation. These ice-nucleating bacteria can influence when and where rain and snow fall, essentially serving as microscopic weather modifiers. The relationship between atmospheric microbes and weather patterns represents one of the most far-reaching examples of microbial architecture. A bacterial spore released from a forest canopy in one continent might eventually seed a cloud that produces rain on another continent. Demonstrating the truly global nature of microbial construction work, recent research has revealed that some plant pathogens have evolved to manipulate atmospheric processes for their own benefit. These bacterial architects cause plant diseases that weaken their hosts, then release themselves into the atmosphere where they serve as highly effective cloud condensation nuclei. The resulting precipitation brings them back to Earth in new locations, allowing them to colonize fresh hosts and expand their architectural projects to new territories high in the atmosphere. Specialized microbial architects work as recyclers, breaking down organic compounds that have been lifted from the Earth's surface and transforming them into new chemical forms. These atmospheric processors handle everything from plant pollen and leaf fragments to industrial pollutants and urban dust. The upper atmosphere presents unique challenges for these microbial architects. Intense ultraviolet radiation from the sun creates a harsh environment where DNA damage is common and cellular repair mechanisms work overtime. Temperatures can fluctuate wildly from blazing heat in direct sunlight to bitter cold in the shadow of clouds. Water is scarce. It's existing only in brief moments when a microbe finds itself inside a cloud droplet. Yet atmospheric microbes have developed remarkable adaptations to thrive in this aerial construction zone. Some produce UV protective pigments that shield their cellular machinery from radiation damage. Others enter dormant states during the harshest conditions, essentially putting their construction projects on hold till more favorable conditions return. These atmospheric architects play crucial roles in global biogeochemical cycles. Nitrogen, sulfur, and carbon compounds as they drift through the sky. They help to clean the atmosphere of pollutants, transform natural organic compounds, and maintain the chemical balance of the air we breathe. One of the most remarkable aspects of atmospheric microbial architecture is the vast distances over which these construction projects operate. Microbes released from the surface can travel thousands of miles on jet streams and trade winds, essentially commuting across continents and oceans to work on construction projects in distant lands. Dust storms in the Sahara Desert lift billions of bacteria and fungi into the atmosphere, carrying them across the Atlantic Ocean to fertilize soils in the Amazon rainforest. These transcontinental construction teams provide essential nutrients, particularly phosphorus, that support the productivity of one of the world's most important ecosystems. Similarly, volcanic eruptions can blast microbial architects high into the stratosphere, where they may circle the globe multiple times before settling back to Earth's surface. These long-distance travelers carry genetic information and construction techniques between widely separated ecosystems, helping to maintain the global connectivity of microbial communities. The seasonal migrations of atmospheric microbes follow predictable patterns linked to weather systems and global circulation patterns. Spring dust storms, summer convection, and winter jet stream patterns all influence the movement of these aerial architects, creating an annual cycle of microbial construction activity that spans the entire planet as we continue our gentle journey. Through the world of unseen architects, it is time to turn our attention inward, to the microscopic builders that live not around us, but within us. The human body, far from being a fortress isolated from the microbial world, is actually a carefully constructed ecosystem, a living collaboration between human cells and trillions of microbial partners who help build and maintain our health, our immunity, and even our behavior. Consider for a moment that in your body at this very moment, microbial cells outnumber human cells by a significant margin. You are not merely a human being carrying some bacteria. You are a walking ecosystem, a mobile biosphere that hosts hundreds of species of microscopic architects, each contributing their unique skills to the grand construction project that is your living, breathing, thinking body. These internal architects begin their work from the moment of birth, and in many ways their construction activities continue throughout your entire life. They build chemical factories in your intestines, construct immune system components, manufacture vitamins and other essential compounds, and even participate in the construction of neural pathways that influence your thoughts and emotions in the warm, dark chambers of your digestive system. Perhaps the most industrious of your internal microbial architects ply their trade. The human gut microbiome represents one of the most complex and productive microbial ecosystems on Earth, rivaling tropical rainforests in its diversity and far exceeding them in its density of living organisms. These intestinal architects work in a carefully organized assembly line, each species specialized for particular aspects of the digestive construction process. Some bacteria excel at breaking down complex carbohydrates that human enzymes cannot process, transforming dietary fiber into short-chain fatty acids that nourish the cells lining your intestinal walls. Others specialize in protein metabolism, amino acid construction, or the synthesis of essential vitamins. The architecture of the gut microbiome is not random, but follows sophisticated organizational principles. Different bacterial species occupy distinct neighborhoods along the length of the intestinal tract, each adapted to the specific chemical and physical conditions of their chosen habitat. The stomach's acid-loving architects differ dramatically from the alkalite-tolerant builders of the small intestine, which in turn differ from the anaerobic contractors of the large intestine. These digestive architects work around the clock, processing every meal you eat and extracting nutrients that would otherwise be lost. They construct a living barrier that protects against harmful microbes, manufacture compounds that reduce inflammation, and even communicate with your immune system to help calibrate your body's defensive responses. The stability and health of this internal ecosystem depends on maintaining the right balance among different microbial contractors. When this balance is disrupted by antibiotics, illness, stress, or dietary changes, the construction projects of your gut microbiome can be thrown into chaos, leading to digestive problems, immune dysfunction, and even changes in mood and behavior on the surface of your body. A different team of microbial architects works to construct and maintain your first line of defense against the outside world. Your skin, the largest organ of your body, hosts a diverse community of bacteria, fungi, and other microorganisms that help build and maintain the protective barrier between your internal environment and the external world. These skin architects work in conditions that would challenge even the hardiest microbes. The surface of human skin is relatively dry, salty, and acidic conditions that would kill many microorganisms. Yet your resident skin microbes have adapted to thrive in this challenging environment. Using specialized metabolic pathways to extract energy and nutrients from the limited resources available, different regions of your skin host different architectural specialists, each adapted to the unique conditions of their particular location. The bacteria that builds communities in the moist, warm environment of your armpits differ significantly from those that construct colonies on the dry skin of your forearms or the oil-rich landscape of your face. These skin architects perform essential construction and maintenance work that helps keep your body healthy. They produce antimicrobial compounds that prevent harmful bacteria from establishing footholds. They help maintain the proper pH balance of your skin. They even influence wound healing and tissue repair processes. The communities of skin microbes also play important roles in your social interactions, though this happens largely below the level of conscious awareness. Each person carries a unique signature of microbial architects, and these microscopic communities contribute to body odor, pheromone production, and other chemical signals that influence how we perceive and interact with one another. Perhaps the most surprising recent discovery about our internal microbial architects is their ability to influence the construction and function of our nervous systems. The gut-brain axis, as scientists call it, represents a complex communication network through which intestinal microbes can influence brain chemistry, behavior, and even personality traits. These neural architects work through multiple pathways to influence brain function. Some produce neurotransmitters, the chemical messengers that brain cells use to communicate with one another. Others manufacture compounds that can cross the blood-brain barrier and directly influence neural activity. Still others communicate with the brain through the vagus nerve, the major communication highway between the gut and the brain. Research has revealed that different species of gut bacteria can influence anxiety levels, depression symptoms, social behavior, and even cognitive performance. These microscopic architects literally help build the chemical foundation for thoughts, emotions, and behaviors, demonstrating that the boundaries between self and microbiome are far more fluid than we once imagined. The implications of this microbial influence on brain function are profound and still being explored. It appears that our internal architects don't just help build and maintain our bodies, they also participate in constructing our minds, our personalities, and our psychological responses to the world around us, as our contemplative journey through the world of unseen architects continues. Let us now expand our view to encompass the truly vast scale at which these microscopic builders operate. Beyond their roles in individual ecosystems, soil formation, and ocean productivity, microbes serve as the primary engineers of our planet's biogeochemical cycles, the global circulation systems that move essential elements through the atmosphere, hydrosphere, lithosphere, and biosphere. These planetary-scale construction projects, orchestrated entirely by microscopic architects, make life on Earth possible. Without their constant work to circulate carbon, nitrogen, phosphorus, sulfur, and other essential elements, our planet would quickly become a lifeless world of locked-up nutrients and stagnant chemistry. Picture these biogeochemical cycles as vast conveyor belts that circle the globe, moving chemical building blocks from one ecosystem to another, from one reservoir to another, from one generation of life to the next. Every turn of these great wheels depends on the metabolic activities of microbes, making these tiny organisms the true drivers of planetary chemistry, the global carbon cycle. Perhaps the most important of these planetary construction projects operates through the coordinated efforts of countless microbial architects working in every environment on Earth. These carbon specialists have been managing the planet's carbon budget for billions of years, long before humans existed, and certainly long before we began to influence atmospheric chemistry through fossil fuel combustion in terrestrial ecosystems. Soil bacteria and fungi serve as the primary carbon processors, breaking down dead plant and animal matter and either releasing the carbon back to the atmosphere as carbon dioxide or incorporating it into stable soil organic matter. The balance between these two processes, decomposition and sequestration, helps determine whether an ecosystem serves as a carbon source or a carbon sink. These soil carbon architects work with exquisite sensitivity to temperature, moisture, and nutrient availability. In cool, moist conditions, decomposition proceeds slowly, allowing organic matter to accumulate in soils and effectively storing carbon underground. In warm, dry conditions, microbial activity accelerates, releasing stored carbon back to the atmosphere. Climate change is altering the working conditions for these soil carbon architects, potentially accelerating decomposition rates and releasing vast stores of soil carbon to the atmosphere. Understanding and predicting the responses of these microscopic builders to changing environmental conditions represents one of the most important challenges in climate science. In marine environments, different teams of carbon architects manage the ocean's role in the global carbon cycle. Photosynthetic microbes in surface waters capture atmospheric carbon dioxide and incorporate it into organic matter, while bacterial decomposers in deep waters break down sinking organic matter and release carbon dioxide back to seawater. The efficiency of this marine carbon pump depends on the activities of countless microscopic architects. From the phytoplankton that carry out primary photosynthesis to the bacteria that process organic matter in deep ocean sediments, changes in ocean temperature, acidity, and nutrient availability can all influence the work of these marine carbon builders, potentially altering the ocean's capacity to absorb and store atmospheric carbon dioxide. The global nitrogen cycle represents perhaps the most complex of the planetary construction projects managed by microbial architects. Nitrogen is essential for all life forms. It's a key component of proteins, DNA, and many other biological molecules. Yet the vast majority of nitrogen on Earth exists in a form that most organisms cannot use. Atmospheric nitrogen gas makes up 78% of the air we breathe. But this nitrogen is locked in an extremely stable triple bond that requires enormous energy to break. Only specialized microbial architects possess the metabolic machinery necessary to capture this atmospheric nitrogen and convert it into forms that other organisms can use. These nitrogen-fixing bacteria, working primarily in soils and aquatic sediments, serve as the entry point for nitrogen into the biosphere. Without their construction work, the biological world would quickly grind to a halt as existing supplies of biologically available nitrogen were depleted, but nitrogen fixation is only the beginning of the microbial nitrogen construction project. Other bacterial specialists, working in different parts of the nitrogen cycle, carry out oxidation and reduction reactions that transform nitrogen between its various chemical forms. Some bacteria specialize in converting ammonia to nitrite, others convert nitrite to nitrate, and still others reduce nitrate back to nitrogen gas. Completing the cycle, this network of nitrogen processing architects operates in a carefully balanced system where the activities of each group influence the others. The nitrogen cycle represents a masterpiece of biological engineering, a global construction project that has been operating smoothly for billions of years and continues to support all life on Earth. Human activities, particularly the industrial production of nitrogen fertilizers and the burning of fossil fuels, have significantly disrupted the work of these nitrogen architects. We have essentially short-circuited their carefully balanced construction system, flooding ecosystems with excess nitrogen and creating imbalances that can persist for decades or centuries. While nitrogen can be captured from the atmosphere, phosphorus, another essential element for all life, exists, primarily in rock minerals that must be slowly weathered and processed before they become biologically available. This makes the microbial architects who specialize in phosphorus processing among the most important builders in terrestrial ecosystems. Phosphorus solubilizing. Bacteria and fungi work directly with rock surfaces, secreting acids and enzymes that can dissolve phosphate minerals and release phosphorus in forms that plants can absorb. These microbial miners operate on geological timescales, slowly liberating phosphorus from its mineral prisons and making it available to the biological world. Other phosphorus architects specialize in recycling this precious element from organic matter, since phosphorus is often the limiting nutrient in terrestrial ecosystems. These recycling specialists play crucial roles in determining ecosystem productivity. They ensure that phosphorus from decomposing plants and animals is quickly returned to forms that living organisms can use. Preventing this essential element from being lost to the ecosystem, the work of phosphorus architects becomes particularly important in highly weathered soils, such as those found in tropical regions, where millions of years of rainfall have leached away most of the icily available phosphorus. In these environments, the efficiency of microbial phosphorus processing often determines whether ecosystems can support lush tropical forests or only sparse vegetation. As we drift deeper into our exploration of unseen architects, let us journey to the most challenging and inhospitable places on our planet, where conditions would seem to make life impossible. Yet even in these extreme environments, places of crushing pressure, scalding heat, bitter cold, intense radiation, or toxic chemistry, microscopic architects have established their workshops and continue their construction projects. These extremophile architects, as scientists call them, push the boundaries of what we consider possible for life. They have evolved construction techniques and metabolic pathways that allow them to thrive in conditions that would instantly kill most organisms. Their work in these extreme environments expands our understanding of life's possibilities and provides insights into how life might exist on other worlds. Far beneath the surface of the Earth, in the hot, high-pressure environment of the deep subsurface, microbial architects work on construction projects that span geological ages. These deep-dwelling builders live in a world of solid rock, where water exists only in tiny fractures and pore spaces, and where nutrients are scarce and energy sources are limited. Some of these subsurface architects derive their energy from the slow radioactive decay of uranium and other radioactive elements in rock minerals. They have learned to harness the radiation that would be lethal to surface-dwelling organisms, using it to power their cellular construction projects. These radiosynthetic microbes represent a completely alternative approach to energy harvesting, demonstrating that life can adapt to use virtually any available energy source. Other deep subsurface architects work with the chemical energy released by reactions between rock, minerals, and groundwater. They serve as living catalysts. Their metabolic activities actually influence the geochemistry of deep groundwater systems and may play roles in processes such as formation and petroleum degradation. The construction projects of these deep earth architects operate on timescales that dwarf human civilization. Some subsurface microbial communities may have been isolated from the surface world for millions of years, yet they continue their slow, patient work of living and reproducing in conditions that seem impossible for life to endure. Research into these deep subsurface architects has revealed that the biomass of microbes living within the earth may rival or exceed the biomass of all surface life. This represents a hidden biosphere, a vast underground realm of microbial construction activity that we are only beginning to understand. In the scalding waters of hot springs, geysers, and underwater volcanic vents, thermophilic architects have constructed some of the most colorful and visually striking microbial communities on Earth. These heat-loving builders thrive at temperatures that would cook most organisms, creating living mats and biofilms that paint hot springs in brilliant oranges, yellows, greens, and reds. The construction work of these thermal architects involves sophisticated adaptations to high-temperature conditions. Their cellular proteins are engineered to remain stable at temperatures that would cause most proteins to unfold and lose their function. Their cell membranes are reinforced with special lipids that maintain their integrity in scalding water. Their DNA repair systems work overtime to fix the damage caused by high-temperature conditions. Different species of thermal architects have specialized for different temperature ranges, creating distinct zones within hot spring systems. The hottest zones closest to the source of geothermal heating host the most extreme thermophiles, bacteria and archaea that can survive temperatures approaching the boiling point of water. Slightly cooler zones support different communities, and the coolest edges of hot springs host more familiar mesophilic organisms. These thermal construction sites provide windows into the possible conditions under which life first evolved on Earth. The hot, chemical-rich environments of geothermal systems may resemble the conditions present on the early Earth. When the first microbial architects began their work of building biological systems from simple, chemical components in the hypersaline environments of salt lakes, salt pans, and underground brine deposits, halophilic architects have mastered the art of construction in conditions of extreme salinity. These salt-loving builders face the constant challenge of maintaining their cellular water balance in environments where the salt concentration would quickly dehydrate most organisms. The adaptations of halophilic architects to high salt conditions are remarkable examples of biological engineering. Some maintain high internal concentrations of salt, essentially matching the salinity of their environment and eliminating the osmotic stress that would otherwise dehydrate their cells. Others accumulate organic compounds that serve as osmotic balancing agents, allowing them to maintain lower internal salt concentrations while still preventing water loss. Many halophilic architects produce distinctive pigments that give salt lakes and salt pans their characteristic pink, red, or purple colors. These pigments serve as protection against the intense solar radiation that often accompanies hypersaline conditions, while also playing roles in the organism's energy metabolism. The construction projects of salt-tolerant architects create some of the most visually striking microbial landscapes on Earth. Salt flats covered with colorful microbial mats, pink salt lakes filled with halophilic algae, and crystalline salt formations encrusted with pigmented bacteria all showcase the artistic sensibilities of these extreme environment specialists. At the opposite extreme of temperature, psychrophilic architects work in the perpetually cold environments of polar regions, high mountain peaks, and deep ocean waters. These cold-adapted builders face the challenge of maintaining cellular activities at temperatures where most biological processes slow to a crawl. The construction techniques of psychrophilic architects involve numerous adaptations to low-temperature conditions. Their enzymes are engineered to remain flexible and active at temperatures that would render most proteins rigid and non-functional. Their cell membranes contain special lipids that remain fluid even in near-freezing conditions. Their metabolic pathways are optimized to function efficiently at low temperatures. In polar ice sheets, microphilic architects work within the crystal structure of ice itself, living in tiny liquid channels and pockets that remain unfrozen even at very low temperatures. These ice-dwelling builders carry out construction projects over extremely long time scales, sometimes remaining active in ice that has been frozen for thousands of years. Permafrost regions host vast communities of cold-adapted architects that remain dormant during the harshest winter conditions but resume their construction activities during brief summer thaws. Climate change is altering the working conditions for these permafrost architects. It's potentially awakening long-dormant construction projects and releasing large quantities of stored carbon to the atmosphere. In our journey through the realm of unseen architects, we have seen how microbes shape landscapes, influence climate, and support ecosystem function through their individual construction projects. But perhaps even more remarkable are the collaborative building projects that emerge when microbes form partnerships with larger organisms, creating integrated structures and systems that neither partner could build alone. These symbiotic architects demonstrate that construction in the biological world often involves cooperation between different types of organisms, each contributing their unique skills and capabilities to joint building projects. The resulting partnerships can be so intimate and well integrated that the individual partners lose their separate identities and function as unified construction teams. Perhaps the most spectacular example of symbiotic architecture can be found in coral reefs, where tiny coral animals work in partnership with microscopic algae to build some of the largest biological structures on Earth. These reef-building collaboratives create underwater cities that can be seen from space, supporting more biodiversity per unit area than any other marine ecosystem. The coral animals serve as the architectural framework, secreting calcium carbonate skeletons that form the physical structure of the reef. But the energy for this massive construction project comes from their microscopic partners. Photosynthetic algae called zooxanthellae that live within the coral tissues and share the products of their photosynthesis with their hosts. This partnership represents a masterpiece of biological integration. The coral provides the algae with a protected environment and access to sunlight, while the algae provide the coral with up to 90% of their energy needs. Neither partner could build reef structures without the other. The coral lacks the energy to construct massive skeletons without photosynthetic support, while the algae lack the ability to create protective calcium carbonate structures. The construction work of coral reef collaboratives creates habitat for countless other organisms. The complex three-dimensional structures built by coral algae partnerships provide shelter, feeding areas, and breeding sites for fish, crustaceans, mollusks, and numerous other marine organisms. Entire ecosystems are built upon the foundation created by these symbiotic architects. Climate change poses serious threats to these collaborative construction projects. Rising ocean temperatures can disrupt the partnership between corals and their algal partners, leading to coral bleaching events where the algae are expelled from coral tissues. Without their photosynthetic partners, the corals cannot maintain their energy-intensive construction work, and reef building grinds to a halt. On land, one of the most successful examples of symbiotic architecture can be found in lichens. Composite organisms, formed by partnerships between fungi and photosynthetic algae or bacteria, these collaborative builders have conquered some of the most challenging terrestrial environments. From Arctic tundra to desert rocks to urban concrete surfaces, in the lichen partnership, the fungal component serves as the primary architect, creating the overall structure and providing protection and water retention capabilities. The photosynthetic partner, either green algae or cyanobacteria, serves as the energy contractor, capturing sunlight and producing organic compounds that fuel the partnership's construction activities. The integration of these partners is so complete that lichens were long considered to be single organisms rather than collaborative partnerships. The fungal architect creates specialized structures to house its photosynthetic partners, while the photosynthetic contractors adjust their metabolism to provide optimal support for the fungal construction activities. Lichen architects are pioneers, in the truest sense, often being the first organisms to colonize bare rock surfaces and begin the long process of soil formation. Their construction activities slowly break down rock surfaces through the production of acids and other chemical weathering agents, creating the initial stages of soil development that will eventually support more complex plant communities. The durability of lichen construction projects is remarkable. Some lichen colonies are hundreds or even thousands of years old, representing some of the longest-lived construction projects on land. Their ability to survive extreme conditions, including prolonged drought, intense cold, and high radiation, makes them valuable indicators of environmental change and air quality beneath the surface of most terrestrial ecosystems. Plant roots and fungal partners collaborate on construction projects that create some of the most extensive networks on Earth. These mycorrhizal partnerships represent one of the most ancient and successful forms of symbiotic architecture, dating back to the earliest colonization of land by plants. In these underground collaboratives, plant roots provide the architectural foundation and the photosynthetic energy that powers construction activities. The fungal partners serve as extension contractors, creating vast networks of thread-like hyphae that can explore soil volumes hundreds of times larger than the root systems. Alone could access the construction techniques employed by these root fungal collaboratives are remarkably sophisticated. Some fungi create elaborate sheathing structures around root tips, forming protective barriers that also serve as nutrient exchange interfaces. Others penetrate directly into root cells, creating intimate cellular-level partnerships, where nutrients and metabolic products are exchanged across living membranes. The networks built by these collaborative architects facilitate communication and resource sharing across entire forest ecosystems. Through their fungal partners, trees can share nutrients with their neighbors, support struggling individuals, and even send chemical warning signals about pest attacks or environmental stresses. Research has revealed that these underground collaborative works display remarkable intelligence-like behaviors. optimally allocating resources, responding to environmental changes, and even showing preferences for supporting closely related individuals. The construction projects of these root fungal collaboratives create what some scientists call the wood wide web, a forest-spanning communication and resource-sharing network as our gentle exploration continues. Let us now consider how the collective work of countless microscopic architects shapes the large-scale landscapes that define our planet's surface. While individual microbes are too small to see, their combined construction activities over geological time have carved valleys, built mountains of sediment, created vast deposits of minerals, and fundamentally altered the geography of continents and ocean basins. These microscopic landscape architects work on timescales that dwarf human civilization, yet their patient construction projects have created many of the geological features that we consider permanent and unchanging. They are the invisible sculptors of Earth's surface, working grain by grain, molecule by molecule, to build the world we inhabit in marine environments. Microscopic architects have been constructing sedimentary deposits for billions of years, creating some of the most extensive geological formations on our planet. These tiny builders, primarily phytoplankton, foraminiferous, and other microscopic organisms with calcium carbonate or silica shells, rain down continuously from surface waters to form vast underwater blankets of biological sediment. The construction work of these marine architects proceeds at a pace measured in millimeters per thousand years. Yet over geological time, their accumulated building projects have created sedimentary layers thousands of meters thick. The famous White Cliffs of Dover, composed primarily of microscopic marine organisms called coccoliths, represent just one example of landscape-scale architecture created entirely by microscopic builders. Different types of microscopic architects create different types of sedimentary constructions. Diatoms, with their intricate glass-like shells, create diatomaceous earth deposits that are mined for industrial uses. Foraminifera, tiny amoeba-like organisms with elaborate calcium carbonate shells, create limestone deposits that can span entire ocean basins. Radiolarians, with their delicate silica skeletons, contribute to chert formations and other siliceous rocks. The timing and location of these microscopic construction projects provide scientists with detailed records of past climate conditions, ocean chemistry, and evolutionary history. Each layer of microbial sediment represents a snapshot of ancient environmental conditions, making these tiny architects invaluable partners in understanding Earth's history. Modern climate change is altering the working conditions for these sedimentary builders, potentially changing the types of sediments being deposited and the rates at which construction proceeds. Ocean acidification, in particular, threatens architects that build calcium carbonate structures, potentially altering sedimentary construction projects that have been ongoing for millions of years. Some microscopic architects work on construction projects of such magnitude that they literally build islands and reshape coastlines. Coral reefs, built primarily through the collaborative efforts of coral animals and their microscopic algal partners, create massive structures that alter ocean currents, provide coastal protection, and serve as foundations for entire island systems. The construction techniques employed by these reef builders are remarkably efficient, using dissolved calcium and carbonate ions from seawater to create solid calcium carbonate structures. The building process requires precise control of chemistry and pH, with the microscopic algal partners playing crucial roles in maintaining the chemical conditions necessary for rapid construction over geological time. These reef construction projects have created entire island chains. Many tropical islands are built entirely on foundations of ancient coral reefs, elevated above sea level by geological processes, but still fundamentally products of microscopic architectural work. The Bahama Banks, the Florida Keys, and countless Pacific atolls all represent landscape-scale construction projects, initiated and maintained by microscopic architects. The three-dimensional complexity of reef structures creates unique hydrodynamic environments that influence wave action, current patterns, and sediment transport over vast areas. These microscopic architects don't just build reefs, they engineer entire coastal systems, creating the environmental conditions that support mangrove forests, seagrass beds, and other marine ecosystems. Rising sea levels and ocean acidification pose serious challenges to these island-building architects. If reef construction cannot keep pace with rising seas, or if acidifying oceans make calcium carbonate construction too difficult, entire island systems and coastal protection structures could be lost within decades or centuries. On land, microscopic. Architects work over thousands of years to construct the layered soil profiles that support terrestrial ecosystems. These patient builders transform solid rock into the structured, nutrient-rich soils that serve as the foundation for plant communities and ultimately all terrestrial life. The construction process begins with pioneer bacterial and fungal architects that colonize bare rock surfaces, secreting acids and other compounds that begin the chemical weathering process. These initial contractors work in partnership with physical weathering agents, like freeze-thaw cycles and thermal expansion, to create the first cracks and fissures in solid rock. As weathering proceeds and organic matter begins to accumulate, different teams of microbial architects take over the construction project. Some specialize in breaking down organic matter and creating humus, the dark, nutrient-rich organic component of soil. Others focus on mineral processing, transforming primary rock minerals into the clay minerals and other secondary minerals that give soil its unique properties. The layered structure of mature soils reflects the organized nature of these microbial construction projects. Different depths host different communities of architects, each adapted to the specific chemical and physical conditions at their chosen level. This vertical organization creates distinct soil horizons with different colors, textures, and chemical properties. The work of soil profile engineers proceeds at different rates in different environments. In tropical regions with warm temperatures and abundant moisture, microbial construction activities can create deep, highly weathered soils over relatively short geological time periods. In cold or arid regions, soil construction proceeds much more slowly, sometimes requiring tens of thousands of years to create thin soil profiles. As we continue our tranquil journey through the world of unseen architects, it becomes clear that these microscopic builders serve not only as constructors of landscapes and ecosystems, but as the primary regulators of our planet's climate system. Through their metabolic activities, their influence on biogeochemical cycles, and their interactions with the atmosphere and oceans, microbes serve as planetary thermostats, helping to maintain the delicate balance of conditions that makes Earth habitable. These climate architects operate on multiple timescales simultaneously, responding to daily and seasonal changes, while also participating in regulatory systems that operate over centuries, millennia, and geological ages. They are both the guardians of climate stability and the agents of climate change. Their collective activities determining whether our planet remains within the narrow range of conditions suitable for complex life. The composition of Earth's atmosphere, the precise mixture of oxygen, carbon dioxide, nitrogen, and trace gases that makes our planet habitable is actively maintained by the metabolic activities of microscopic architects. These atmospheric managers work around the clock to process gases, regulate concentrations, buffer against rapid changes that could disrupt the climate system. Photosynthetic microbes in both terrestrial and marine environments serve as the primary sources of atmospheric oxygen, continuously replenishing this essential gas through their light harvesting construction work. Without their constant production, atmospheric oxygen levels would gradually decline as the gas is consumed by respiration, weathering reactions, and other processes. The balance between photosynthetic oxygen production and respiratory oxygen consumption is maintained by vast networks of microbial architects working in different environments. Changes in temperature, nutrient availability, or other environmental factors can alter the activities of these atmospheric managers, potentially shifting the global balance between oxygen production and consumption. Carbon dioxide levels in the atmosphere are similarly regulated by microbial activities. Marine phytoplankton remove carbon dioxide from both the atmosphere and surface ocean waters. While soil bacteria and fungi release carbon dioxide through their decomposition work, the balance between these processes helps determine atmospheric carbon dioxide concentrations and, consequently, global climate conditions. Other trace gases in the atmosphere are also processed by specialized microbial architects. Methane oxidizing. Bacteria consume atmospheric methane, helping to regulate levels of this potent greenhouse gas. Nitrogen processing bacteria influence atmospheric nitrous oxide concentrations, while sulfur metabolizing microbes affect atmospheric sulfur compounds that influence cloud formation and climate, the thermal properties of the oceans there. Capacity to absorb, store, and transport heat around the globe are significantly influenced by the activities of marine microbial architects. These oceanic climate regulators affect everything from local water temperatures to global circulation patterns that distribute heat between equatorial and polar regions. Marine phytoplankton influence. Ocean temperatures, through their effects on light absorption and heat distribution, dense blooms of microscopic algae can alter the optical properties of surface waters, changing how much solar energy is absorbed versus reflected back to space. Different species of phytoplankton have different effects on ocean optics, making the composition of marine microbial communities an important factor in regional climate regulation. The vertical distribution of marine microbes also affects ocean temperature profiles. Some bacteria and archaea specialize in processing organic matter at specific depths, influencing the distribution of nutrients and the formation of distinct water layers with different temperatures and densities. These layered structures affect ocean circulation patterns and heat transport. Dimethyl sulfide DMs, produced by certain marine phytoplankton, plays important roles in cloud formation over the oceans. This microbially produced compound can influence the formation and properties of marine clouds, affecting how much solar energy reaches the ocean's surface and how much is reflected back to space. Changes in marine microbial communities can thus influence regional and global climate through their effects on cloud formation. Ocean acidification, caused by absorption of excess atmospheric carbon dioxide, is altering the working conditions for marine climate architects. Changes in ocean chemistry can affect the survival and activities of different microbial species, potentially disrupting climate regulation systems that have operated for millions of years. Perhaps the most sophisticated aspect of microbial climate architecture lies in the feedback systems that these tiny builders create and maintain. These regulatory networks allow the climate system to respond to changes and maintain stability over long time periods. Much like a thermostat maintains steady temperature in a building, one of the most important climate feedback systems involves the relationship between temperature and microbial activity in soils. As temperatures rise, soil bacteria and fungi generally become more active, decomposing organic matter more rapidly and releasing more carbon dioxide to the atmosphere. This creates a positive feedback loop where warming leads to increased carbon dioxide emissions, which leads to more warming. However, other microbial feedback systems work in the opposite direction, providing negative feedback that tends to stabilize climate conditions. Increased atmospheric carbon dioxide can stimulate plant growth, which provides more organic matter for soil microbes and can lead to increased carbon storage in soils. Marine phytoplankton may respond to warming by increasing their production of cloud-forming compounds, creating more reflective clouds that cool the surface. The interactions between different microbial feedback systems create complex regulatory networks that scientists are still working to understand. These networks involve multiple types of architects working in different environments. With feedback loops operating on timescales from days to millennia, climate change is testing these microbial regulatory systems in unprecedented ways. The rapid pace of current environmental change may exceed the response capacity of some feedback systems, potentially leading to climate regulation failures and rapid transitions to new climate states. As our contemplative journey through the microbial world draws toward its gentle conclusion, let us consider perhaps the most profound construction project undertaken by these unseen architects: the building of evolution itself. Microbes are not merely the products of evolutionary processes, They are active participants in shaping the direction and pace of evolution, creating the environmental conditions and selective pressures that determine how life develops and diversifies over geological time. These evolutionary architects work through multiple mechanisms to influence the development of life on Earth. They create new ecological niches, establish symbiotic partnerships, transfer genetic material between distant lineages, and modify environmental conditions in ways that open new evolutionary pathways and close others. Microbial architects are master builders of ecological niches. The environmental conditions and resource availability, patterns that determine which organisms can survive and thrive in particular locations. Through their metabolic activities, these tiny builders create chemical gradients, modify pH conditions, alter oxygen levels, and generate countless other environmental variables that create opportunities for specialized organisms. The construction of new ecological niches by microbes often leads to evolutionary innovation. As organisms adapt to exploit these newly available environmental opportunities, for example, oxygen produced by early photosynthetic bacteria created entirely new niches for oxygen-utilizing organisms, leading to the evolution of aerobic respiration and all the complex life forms that depend on this highly efficient energy processing system. Similarly, the acidic conditions created by sulfur-oxidizing bacteria in mining environments have led to the evolution of acid-tolerant organisms with novel biochemical adaptations. The high salt conditions maintained by halophilic archaea create niches for salt-adapted organisms with unique cellular mechanisms. For dealing with osmotic stress, these niche construction activities create cascading effects throughout evolutionary history. As new environmental conditions are established by microbial architects, new evolutionary pressures emerge, leading to adaptive radiation and the development of new lineages. The organisms that evolve in these constructed niches often develop intimate relationships with their microbial architects, creating coevolutionary partnerships that drive further innovation. The ongoing construction of new niches by microbial architects continues to drive evolutionary innovation today. Human activities have created new environments, polluted sites, urban ecosystems, industrial facilities that are rapidly being colonized by microbial architects, who modify these novel conditions and create opportunities for evolutionary adaptation. Unlike most larger organisms, microbial architects routinely engage in horizontal gene transfer, the direct sharing of genetic material between individuals that may be distantly related or even belong to different species. This genetic construction work allows evolutionary innovations to spread rapidly through microbial communities and even jump between major lineages of life. These horizontal construction contractors serve as genetic engineers, moving useful genetic modules between organisms and allowing rapid adaptation to new environmental challenges. Antibiotic resistance genes, for example, can spread rapidly through bacterial communities via horizontal transfer, allowing multiple species to quickly develop. Resistance to new antimicrobial compounds, the genetic construction work performed by these microbial architects has contributed essential capabilities to virtually all forms of life. Many of the fundamental biochemical pathways used by plants, animals, and fungi were originally developed by bacterial architects and later transferred to other lineages through various mechanisms. Photosynthesis. One of the most important biological innovations in Earth's history spread from its bacterial origins to algae and plants through horizontal transfer events. The chloroplasts that carry out photosynthesis in plant cells are actually descendants of ancient bacterial architects that took up residence inside larger cells and eventually became permanent cellular components. This horizontal construction work continues to play important roles in modern evolutionary processes. Environmental changes can create strong selective pressures for particular genetic innovations, and horizontal transfer allows these innovations to spread rapidly through microbial communities and potentially to other organisms as well. Many of the most important evolutionary innovations in the history of life have emerged from partnerships between microbial architects and larger organisms. These symbiotic developers create intimate associations that benefit both partners and often lead to evolutionary innovations that neither partner could achieve alone. The evolution of complex cells with nuclei and other internal structures represents one of the most important partnership development projects in evolutionary history. According to endosymbiotic theory, these complex cells evolved when bacterial architects took up residence inside larger cells, eventually becoming specialized cellular components like mitochondria and chloroplasts. These ancient partnership development projects created the cellular architecture that makes complex multicellular life possible. Without the energy processing capabilities provided by mitochondrial descendants of bacterial architects, large multicellular organisms could not exist. Without the photosynthetic capabilities provided by chloroplast descendants of bacterial architects, plants could not exist. More recent partnership development projects have led to numerous evolutionary innovations in multicellular organisms. The evolution of ruminant digestion, which allows cows, sheep, and other herbivores to digest cellulose, depends entirely on partnerships with specialized microbial architects that live in their digestive systems. Similarly, the evolution of bioluminescence in many marine organisms involves partnerships with light-producing bacterial architects. The evolution of nitrogen fixation capabilities in certain plants depends on partnerships with nitrogen-fixing bacterial architects. The evolution of defensive capabilities in many organisms involves partnerships with antibiotic-producing microbial architects. These symbiotic partnership development projects continue to drive evolutionary innovation today. Environmental changes create new selective pressures that favor organisms capable of forming beneficial partnerships with microbial architects, leading to the evolution of new symbiotic associations and the capabilities they provide. As our gentle journey through the realm of unseen architects completes its circle, we return to where we began, to the recognition that these microscopic builders are not merely components of the world around us, but the fundamental constructors of the very possibility of our existence. In every breath we take, in every step we make upon the soil, in every drop of water we drink, we encounter the handiwork of these patient, invisible architects who have been building our world for billions of years. We began this contemplative exploration by imagining ourselves shrinking down to walk among these tiny builders, and now, as we prepare to return to our human scale of existence, we carry with us a new understanding of the vast construction projects that surround us always. The air that fills our lungs was crafted by photosynthetic architects working in forests and oceans. The soil that supports our food was built grain by bacterial and fungal contractors. The climate that maintains our planet's habitability is regulated by countless microbial thermostats working around the clock. As you settle into sleep, dear listener, remember that the construction work of these unseen architects never ceases. Even now, as your consciousness fades and your body rests, billions of microbial builders continue their work both around you and within you. In the soil beneath your bed, bacterial architects are processing organic matter and cycling nutrients. In the air around you, atmospheric architects are processing gases and helping to regulate the composition of the atmosphere you breathe within your own body. Internal microbial architects work through the night, maintaining your digestive system, supporting your immune function, and even influencing your dreams through their effects on brain chemistry. You are never alone. You are always surrounded by and filled with these microscopic partners who contribute to every aspect of your existence. The construction projects of these unseen architects operate on time scales that span from microseconds to geological ages. Some bacterial reactions proceed thousands of times per second, while other microbial construction projects unfold over millions of years. Yet all of these different timescales are integrated into the vast, interconnected system of construction activity that maintains our living planet. This eternal construction site is not random or chaotic, but follows principles of organization and cooperation that scientists are only beginning to understand. The invisible cities built by microbes are more complex than human cities, more interconnected than our global communication networks. It's more efficient than our industrial systems, and more sustainable than any technology we have yet devised. In Contemplating the work of these unseen architects, we encounter profound lessons about cooperation, sustainability, and the power of small actions accumulated over time. Individual microbes are tiny and seemingly insignificant, yet their collective efforts have transformed an entire planet and made complex life possible. They demonstrate that the greatest constructions emerge not from the work of single powerful builders, but from the coordinated efforts of countless small contributors. These microscopic architects teach us patience. Their construction projects unfold over timescales that make human endeavors seem brief and fleeting. They remind us that the most important work often happens slowly, steadily, without fanfare or recognition. The soil beneath a forest has been under construction for thousands of years, each generation of microbial builders adding their contributions to a project that began long before they existed and will continue long after they are gone. They teach us about resilience and adaptation. Faced with changing conditions, environmental challenges, and even catastrophic events, these invisible architects find ways to continue their work. They develop new construction techniques, form new partnerships, and discover new sources of energy and materials. They demonstrate that survival depends not on rigid adherence to old methods, but on the flexibility to innovate and adapt while maintaining essential functions. Most importantly, they teach us about interconnectedness. No microbial architect works in isolation. Each depends on the construction work of others. Each contributes to the success of the whole community. And each plays multiple roles in the vast web of relationships that makes life possible. They show us that individual success and community well-being are not separate goals, but aspects of the same fundamental principle of mutual support and cooperation. As our world changes, as climate shifts, as human activities alter landscapes, and ocean chemistry, as new pollutants and challenges emerge, the unseen architects adapt their construction projects to meet new conditions. They are not passive victims of environmental change, but active responders who modify their building techniques, form new partnerships, and develop new solutions to emerging challenges in polluted environments. Bacterial architects evolve new metabolic pathways that can break down synthetic chemicals and transform toxic compounds into harmless byproducts in acidifying oceans. Marine architects develop new shell-building techniques that can function under changing chemical conditions. In warming soils, fungal architects adjust their construction schedules and modify their partnerships with plants to maintain ecosystem function under thermal stress. These future architects are already at work, preparing construction projects that will help life adapt to the changing conditions of the Anthropocene. They remind us that life is not fragile, but remarkably resilient and creative. While individual species may struggle with environmental change, the microbial architects collectively possess the genetic diversity, metabolic flexibility, and evolutionary creativity to develop solutions to almost any challenge or changing world might present. Understanding and working with these future architects represents one of our greatest opportunities for addressing environmental challenges. Rather than fighting against natural processes, we can learn to partner with microbial architects, supporting their construction work and harnessing their capabilities to address problems like climate change, pollution, and ecosystem degradation. Bioremediation. Projects use bacterial architects to clean up contaminated sites. Carbon sequestration efforts support soil architects in their work of storing carbon in terrestrial ecosystems. Sustainable agriculture practices nurture the microbial architects that maintain soil health and plant nutrition. These human microbial partnerships represent a new phase in the long history of construction collaboration on Earth. Let us delve even deeper into the microscopic realm, to the level where individual molecules dance and quantum effects influence the construction work of our unseen architects. Here, at the intersection of biology and physics, microbial builders employ construction techniques that operate according to the strange rules of quantum mechanics, where particles can exist in multiple states simultaneously and where the very act of observation can change the outcome of construction processes. These quantum architects work with tools and techniques that seem almost magical when viewed from our macroscopic perspective. They manipulate individual atoms, orchestrate precise electron transfers, and harness quantum coherence effects to achieve construction efficiencies that exceed anything human technology has yet accomplished. Perhaps the most remarkable quantum architects are the photosynthetic microbes that have mastered the art of capturing and converting light energy with near-perfect efficiency. These tiny engineers employ quantum mechanical effects to guide energy through their molecular construction machinery in ways that maximize energy capture and minimize energy loss. When sunlight strikes a photosynthetic bacterial cell, it encounters an incredibly sophisticated quantum construction system. The light harvesting complexes in these cells contain precisely arranged molecules that can maintain quantum coherence, a state where energy exists in multiple pathways simultaneously, till the most efficient route is selected for energy transfer. This quantum construction work allows photosynthetic architects to achieve energy conversion efficiencies approaching the theoretical maximum. They waste virtually no energy in their light harvesting operations, capturing nearly every available photon and converting its energy into useful chemical bonds with remarkable precision. The molecular architecture of these quantum construction systems is breathtakingly complex. Individual chlorophyll molecules are positioned with atomic-level precision, their spacing and orientation optimized to facilitate quantum energy transfer processes. Accessory pigments are arranged in carefully designed arrays that funnel energy toward reaction centers where photochemical construction work takes place. These quantum architects have been perfecting their construction techniques for billions of years, long before human physicists discovered quantum mechanics or understood the principles that govern molecular-level energy transfer. They represent master engineers whose construction capabilities far exceed our current technological abilities throughout the microbial world. Enzymatic architects employ quantum tunneling effects and other quantum mechanical phenomena to carry out construction work with extraordinary precision and speed. These molecular contractors can manipulate individual chemical bonds, position atoms with sub-nanometer accuracy, and catalyze reactions that would be impossible without quantum mechanical assistance. Enzyme Times architects work by creating precisely shaped molecular construction sites that bring reactant molecules into optimal positions for bond formation or breaking. The construction process often involves quantum tunneling, where particles pass through energy barriers that would be impossible to overcome using classical physics alone. This allows enzymatic architects to carry out construction work at rates that can exceed 1 million operations per second. The precision of enzymatic construction work is almost unimaginable. These molecular architects can distinguish between chemical compounds that differ by only a single atom, select specific stereoisomers from mixtures of nearly identical molecules, and carry out multi-step construction sequences with error rates approaching zero. Different types of enzymatic architects specialize in different aspects of molecular construction. Some excel at building large molecules from smaller components, working like molecular assembly lines to construct complex proteins, nucleic acids, and other biological polymers. Others specialize in demolition work, breaking down large molecules into their component parts with surgical precision. The construction techniques employed by enzymatic architects often involve conformational changes, alterations in the shape of the enzyme molecule that occurred during the construction process. These shape changes allow the enzyme to bind reactants, facilitate chemical reactions, and release products in carefully orchestrated sequences that maximize construction efficiency and accuracy at the quantum level. Microbial architects face unique challenges in constructing cellular membranes that must be simultaneously permeable and selective, allowing essential molecules to pass while excluding harmful compounds. These membrane specialists employ quantum mechanical effects to create molecular scale gates, channels, and transport systems that operate with remarkable sophistication and precision. The construction of functional cellular membranes requires precise control over molecular interactions at the quantum level. Lipid molecules must be arranged in bilayer structures that maintain appropriate fluidity while providing effective barriers. Membrane proteins must be inserted with correct orientations and conformations to function as molecular machines for transport, signaling, and energy conversion. Ion channel architects create molecular construction projects that allow specific ions to pass through cellular membranes while excluding others. These channels operate through quantum mechanical selectivity, filters that can distinguish between ions of different sizes, charges, and quantum properties. The construction work is so precise that channels specific for sodium ions will exclude potassium ions, despite the fact that these ions differ in size by only a fraction of an angstrom. Transport protein. Architects construct molecular machines that use energy from various sources to move molecules against concentration gradients. These construction projects often involve conformational changes that occur in response to quantum mechanical interactions with specific molecules, creating molecular construction processes that can operate in reverse, using concentration gradients, to generate energy for other cellular construction work. As we continue our gentle exploration of the unseen architectural realm, let us consider how these microscopic builders operate across the vast expanse of time, creating construction projects that span everything from nanoseconds to geological epochs. These temporal architects demonstrate that construction in the microbial world is not confined to spatial dimensions, but extends through time itself, creating structures and systems that persist, evolve, and adapt across multiple temporal scales. Simultaneously within individual microbial cells, circadian architects maintain internal construction schedules that coordinate cellular activities with the daily cycle of light and darkness. These temporal builders construct molecular clockwork mechanisms that operate with remarkable precision, allowing cells to anticipate environmental changes and optimize their construction activities for different times of day. The construction work of circadian architects involves complex networks of interacting proteins that oscillate in concentration and activity over 24-hour cycles. These molecular timekeeper systems coordinate everything from DNA replication and cell division to energy metabolism and stress response systems, ensuring that different types of construction work occur at optimal times. Marine photosynthetic architects, for example, use circadian construction coordination to time their light harvesting activities with periods of maximum sunlight availability. They construct and deconstruct light harvesting complexes on daily schedules, building up their photosynthetic capacity during daylight hours and reallocating resources to other construction projects. During darkness, soil bacterial architects coordinate their construction activities with daily temperature and moisture cycles, adjusting their metabolic rates and construction schedules to match environmental conditions. This temporal coordination allows them to maximize construction efficiency while minimizing energy waste and cellular stress. The precision of circadian construction coordination is remarkable, with some microbial architects maintaining accurate 24-hour schedules even when isolated from external time cues. These internal chronometers represent some of the most sophisticated temporal construction systems in the biological world, demonstrating that time itself can serve as a construction material for organizing cellular activities. Beyond daily cycles, many microbial architects coordinate their construction projects with seasonal patterns, adjusting their activities to match annual cycles of temperature, precipitation, nutrient availability, and ecosystem productivity. These seasonal planners demonstrate the ability to anticipate long-term environmental changes and modify their construction schedules accordingly. In temperate soils, bacterial and fungal architects prepare for winter by modifying their cellular construction projects to increase cold tolerance and reduce metabolic rates. They accumulate protective compounds, strengthen cell walls, and shift their metabolism toward more efficient pathways that can function at low temperatures. Spring awakening involves massive construction projects as soil architects emerge from winter dormancy and begin processing the accumulated organic matter from fallen leaves and dead plant material. The timing of these spring construction activities is carefully coordinated with plant growth cycles, ensuring that nutrients are released when growing plants need them most. Aquatic microbial architects coordinate their construction activities with seasonal patterns of water temperature, ice formation, and nutrient mixing. In lakes and oceans, seasonal construction planning involves preparing for thermal stratification, planning for spring and fall mixing events, and adjusting construction schedules to match the seasonal availability of nutrients and light. The construction projects of seasonal planners often involve community-level coordination, with different species of microbial architects timing their activities to complement rather than compete with each other. This temporal niche partitioning allows diverse microbial communities to coexist and contribute to ecosystem function throughout the annual cycle. At the longest temporal scales, microbial architects participate in construction projects that span millions of years, gradually transforming planetary environments and creating the conditions that allow evolution to proceed in new directions. These geological time constructors work at rates so slow that their progress is nearly imperceptible, yet their cumulative effects reshape continents and alter global climate systems. The construction of major geological formations, limestone beds, coal deposits, oil reservoirs, and other sedimentary structures, represents the accumulated work of countless generations of microbial architects. Each individual organism contributes only a microscopic amount to these projects, yet their collective efforts over geological time create formations that can be hundreds of meters thick and extend across entire continents. These geological constructors demonstrate patience and persistence on scales that dwarf human comprehension. The bacterial architects that contribute to limestone formation work at rates measured in fractions of millimeters per century. Yet their construction projects have created massive carbonate platforms that influence ocean circulation patterns and global climate systems. The weathering of rock surfaces by microbial architects proceeds at similarly slow rates. Yet over geological time, these construction activities have created the soil profiles that support terrestrial ecosystems. The patient work of rock-dwelling bacteria and fungi, secreting acids and other weathering agents, gradually transforms solid bedrock into the layered soil systems that make plant life possible. Climate regulation by microbial architects operates on multiple geological timescales simultaneously. Short-term regulation involves daily and seasonal adjustments in microbial activity that buffer against rapid environmental changes. Long-term regulation involves gradual shifts in microbial communities and construction activities that help maintain planetary habitability over millions of years. In the quiet depths of our exploration, we encounter perhaps the most profound construction work undertaken by unseen architects: the building and maintenance of biological information systems. These microscopic librarians and information engineers construct, preserve, and transmit the genetic libraries that contain the blueprints for all life on Earth, while also creating sophisticated communication networks that allow microbial communities to share information and coordinate their construction activities. Within every microbial cell, molecular architects work continuously to maintain vast libraries of genetic information. These library builders face the monumental challenge of preserving information stored in DNA molecules while also allowing for the controlled modifications that drive evolution and adaptation. Their construction work must balance stability with flexibility, ensuring that essential genetic information is preserved while allowing for the innovations that enable adaptation to changing conditions. The construction techniques employed by genetic library builders operate with extraordinary precision. DNA polymerases, the molecular architects responsible for copying genetic information, work with error rates approaching 1 mistake per billion operations. When errors do occur, specialized repair architects immediately identify and correct most mistakes. Maintaining the integrity of the genetic library across countless generations, these molecular librarians organize genetic information using sophisticated indexing systems that allow rapid access to specific genes when their products are needed. Regulatory architects construct complex control networks that determine which genes are active under different conditions, essentially creating dynamic library systems where different collections of books are made available depending on current construction needs. The preservation of genetic libraries across geological time represents one of the most remarkable construction achievements in the history of life. Some genetic sequences have been maintained with remarkable fidelity for millions of years, while others have been modified and improved through evolutionary processes. The balance between conservation and innovation in these genetic construction projects has allowed life to persist through dramatic environmental changes, while continuously developing new capabilities. Horizontal gene transfer allows microbial architects to share genetic construction, blueprints between different organisms, and even different species. This genetic construction sharing creates vast distributed libraries where useful genetic innovations can spread rapidly through microbial communities, accelerating adaptation and allowing collective responses to environmental challenges. Beyond their individual cellular libraries, microbial architects construct sophisticated chemical communication networks that allow them to coordinate their construction activities across entire communities. These chemical communication engineers develop and maintain signaling systems that can convey complex information about environmental conditions, resource availability, and community needs. Quorum sensing represents one of the most sophisticated chemical communication systems constructed by microbial architects. These systems allow bacterial communities to monitor their population density and coordinate collective responses when their numbers reach critical thresholds. The construction of quorum sensing networks involves the production, detection, and interpretation of chemical signals that convey information about community size and composition. When bacterial architects detect that their community has reached appropriate population densities, they can collectively initiate large-scale construction projects that would be impossible for individual cells to accomplish alone. Biofilm formation, antibiotic production, the secretion of digestive enzymes often require coordinated community action that depends on chemical communication networks. Interspecies chemical communication allows different types of microbial architects to coordinate their construction activities even when they belong to different species or even different kingdoms of life. Plant roots communicate with fungal partners through complex chemical signaling systems that coordinate nutrient exchange and mutual support. Bacterial architects and animal digestive systems communicate with their hosts through chemical signals that influence immune responses and digestive processes. The chemical languages developed by microbial communication engineers are remarkably sophisticated, employing multiple types of signaling molecules to convey different types of information. Some signals indicate the presence of nutrients or energy sources, others warn of environmental dangers, and still others coordinate reproductive activities or collective responses to threats. Perhaps most remarkably, some microbial architects have developed the ability to store and retrieve memories of past experiences, creating biological memory systems that allow them to anticipate future conditions and prepare appropriate responses. These memory storage specialists demonstrate that information processing and learning are not unique to complex nervous systems, but can emerge from the construction activities of individual cells. Bacterial memory systems often involve epigenetic modifications, changes in gene expression patterns that persist after the initial stimulus has disappeared. These modifications serve as molecular memory traces that allow cells to remember past environmental conditions and respond more rapidly when similar conditions are encountered again. Some bacterial architects can remember seasonal patterns, adjusting their construction activities in anticipation of predictable environmental changes. Others can remember the locations of favorable or unfavorable environmental conditions, modifying their movement patterns based on past experiences. Still others can remember previous encounters with toxins or stressors, preparing defensive responses that can be activated more rapidly if similar threats are encountered. The construction of biological memory systems involves complex molecular machinery that can encode, store, and retrieve information about past experiences. These memory architects demonstrate that information processing capabilities can emerge from relatively simple molecular construction projects, suggesting that memory and learning may be fundamental properties of living systems rather than emergent properties of complex brains. Collective memory systems allow microbial communities to store and share information about environmental conditions across multiple generations. The composition and structure of microbial communities themselves serve as a form of ecological memory, preserving information about past environmental conditions and construction strategies that proved successful under different circumstances. As we drift further into our contemplative journey, let us turn our attention to the microbial architects who specialize in constructing survival systems, the microscopic engineers who build resilience, redundancy, and recovery capabilities into biological systems. These survival specialists work continuously to prepare for uncertain futures, constructing flexible systems that can withstand disturbances, adapt to changing conditions, and recover from catastrophic events. When environmental conditions become challenging, specialized microbial architects immediately begin constructing stress response systems designed to protect cellular infrastructure and maintain essential functions under adverse conditions. These stress response constructors work with remarkable speed and efficiency, rapidly deploying protective systems that can mean the difference between survival and death. Heat shock architects respond to temperature stress by constructing protective proteins that prevent cellular damage during thermal extremes. These molecular chaperones work like microscopic emergency response teams, rescuing damaged proteins and helping to rebuild cellular structures that have been disrupted by high temperatures. Their construction work can be completed within minutes of detecting thermal stress, providing critical protection during heat waves and other thermal emergencies. Osmotic stress. Architects construct protective systems that help cells maintain proper water balance when faced with changes in salinity or water availability. These builders rapidly accumulate protective compounds called compatible solutes that help maintain cellular integrity under conditions that would otherwise cause cells to shrivel or burst due to osmotic imbalances. Oxidative stress architects construct antioxidant defense systems that protect cellular components from damage by reactive oxygen species and other harmful chemicals. These protective constructors maintain arsenals of antioxidant molecules and repair enzymes that can neutralize harmful chemicals and repair the damage they cause to cellular infrastructure. The construction of stress response systems often involves rapid changes in gene expression patterns, with stress response architects activating emergency construction programs that prioritize survival over growth or reproduction. These emergency protocols demonstrate the remarkable ability of microbial architects to rapidly reorganize their construction priorities in response to changing conditions, when environmental conditions become too severe for normal construction activities. Specialized microbial architects construct dormancy systems that allow cells to survive extended periods of unfavorable conditions. These dormancy engineers build sophisticated suspended animation systems that can preserve cellular integrity for months, years, or even decades, while consuming minimal energy and resources. Spore-forming architects represent some of the most skilled dormancy engineers in the microbial world. These builders construct elaborate protective structures that can withstand extreme heat, cold, radiation, desiccation, and chemical exposure. The construction of bacterial spores involves the creation of multiple protective layers, specialized DNA, binding proteins, and unique metabolic systems that can remain viable for extraordinary periods of time. Some bacterial spores constructed by ancient dormancy architects have been revived after thousands or even millions of years of dormancy, demonstrating the remarkable durability of these biological construction projects. These long-term survival systems represent some of the most sophisticated preservation technologies ever developed, surpassing anything human engineering has achieved for long-term information and material storage. Cyst-forming architects construct protective structures that allow cells to survive unfavorable conditions while retaining the ability to rapidly resume normal activities when conditions improve. These reversible dormancy systems provide a more flexible approach to survival, allowing rapid awakening when opportunities for growth and reproduction return. The timing of dormancy systems construction is carefully coordinated with environmental cues that indicate approaching unfavorable conditions. Dormancy architects can detect subtle changes in temperature, moisture, nutrient availability, and other environmental factors, allowing them to begin dormancy construction before conditions become life-threatening. After surviving environmental challenges or catastrophic events, specialized microbial architects focus on recovery and repair activities. designed to restore normal cellular function and rebuild damaged infrastructure. These recovery specialists work with remarkable efficiency to assess damage, prioritize repairs, and reconstruct essential systems as quickly as possible. DNA. Repair architects represent some of the most critical recovery specialists in microbial communities. These molecular contractors can detect and repair various types of genetic damage, from simple base substitutions to complex chromosomal rearrangements. Their repair work is essential for maintaining genetic integrity and ensuring that construction blueprints remain accurate across generations. Membrane repair. Architects specialize in rebuilding cellular boundaries that have been damaged by physical or chemical stress. These contractors can patch holes in cellular membranes, replace damaged lipid molecules, and reconstruct membrane proteins that have been inactivated by environmental stress. Their rapid repair work is essential for maintaining cellular integrity and preventing further damage. Metabolic repair architects focus on rebuilding the enzymatic machinery that drives cellular construction activities. These specialists can replace damaged enzymes, reconstruct metabolic pathways that have been disrupted by stress, and rebalance cellular chemistry to restore normal construction capabilities. The coordination of recovery activities involves sophisticated damage assessment systems that can evaluate the extent of injury and prioritize repair activities accordingly. Recovery architects must balance the need for rapid repair against the limited resources available for reconstruction work. It's making complex decisions about which systems to repair first and which can wait for later reconstruction phases. In the gentle rhythm of our continuing exploration, we encounter perhaps the most elegant aspect of microbial architecture: the construction of harmony and balance within complex systems. These harmony builders work not as individual contractors, but as members of vast orchestras, playing their part in maintaining the delicate balance that allows ecosystems to function as integrated wholes throughout every ecosystem on Earth. Microbial architects work as balance engineers, constructing feedback systems and regulatory networks that maintain stability and prevent any single component from dominating the entire system. These balance engineers demonstrate that sustainability emerges not from rigid control, but from flexible networks of mutual interdependence and reciprocal support in forest soils. Balance engineers coordinate the activities of hundreds of bacterial and fungal species, ensuring that nutrient cycling proceeds smoothly and that no single group of decomposers overwhelms the system. They construct chemical communication networks that allow different species to coordinate their activities and avoid destructive competition for limited resources. These soil balance engineers respond to changes in plant composition, seasonal patterns, and environmental conditions by adjusting their construction activities to maintain ecosystem stability. When tree species change the types of organic matter they contribute to the soil, the balance engineers modify their decomposition processes to handle new types of chemical inputs while maintaining overall system function. Marine balance engineers work on even larger scales, coordinating the activities of planktonic communities across entire ocean basins. These oceanic harmony builders construct nutrient cycling systems that can process vast quantities of organic matter while maintaining the chemical balance of seawater and supporting the entire marine food web, the construction work of ecosystem. Balance engineers often involves creating redundancy, multiple pathways for accomplishing essential functions so that the system can continue operating even if individual components fail. This redundant architecture provides resilience against disturbances and ensures that critical ecosystem functions can be maintained under changing conditions within microbial communities. Specialized architects work as metabolic network coordinators, constructing integrated systems where the waste products of some organisms serve as the raw materials for others. These coordinators build circular construction systems that minimize waste and maximize the efficiency of resource utilization across entire communities. In anaerobic environments, metabolic network coordinators construct elaborate chains of chemical transformations where complex organic molecules are gradually broken down through a series of steps, each carried out by different specialist organisms. The final products of this coordinated construction work are simple compounds like methane and carbon dioxide that can be used by other organisms or released to the environment. Centrifugal partnerships represent some of the most sophisticated metabolic coordination projects in the microbial world. These partnerships involve organisms that have evolved such intimate metabolic integration that neither partner can survive without the other. The waste products of one organism are immediately consumed by its partner, creating tightly coupled construction systems with extraordinary efficiency. The coordination of metabolic networks requires precise timing and spatial organization. Network coordinators must ensure that organisms producing specific compounds are located near organisms that consume those compounds, and that production and consumption rates are properly balanced to prevent the accumulation of toxic intermediates. These metabolic coordination projects often involve complex, throwaway or multi-way partnerships where several different organisms contribute different enzymatic capabilities to collective construction processes. The coordination required for these partnerships demonstrates remarkable biochemical communication and cooperation abilities. At the cellular level, microbial architects construct homeostatic systems that maintain stable internal conditions. Despite fluctuating external environments, these homeostatic builders create regulatory networks that can detect changes in cellular conditions and rapidly implement corrective measures to restore balance and stability, pH. Homeostasis architects construct sophisticated buffering systems that maintain optimal acidity levels within cells despite changes in environmental chemistry. These systems involve multiple regulatory mechanisms that can rapidly adjust cellular pH by modifying enzyme activities, transport processes, and metabolic pathways. Temperature homeostasis builders construct adaptive systems that allow cells to function effectively across temperature ranges by adjusting membrane composition, enzyme activities, and metabolic rates. These systems provide both immediate responses to temperature changes and longer-term adaptations to sustained temperature shifts. Nutrient homeostasis architects construct storage and regulatory systems that maintain adequate supplies of essential nutrients while preventing the accumulation of excess materials that could disrupt cellular function. These systems involve complex sensing mechanisms that can detect nutrient levels and adjust uptake and storage activities accordingly. The construction of homeostatic niche systems involves creating multiple layers of regulation, from rapid molecular responses that occur within seconds to slower adaptation processes that unfold over hours or days. This multi-layered architecture provides both immediate protection against environmental fluctuations and longer-term stability under changing conditions. As our journey approaches its gentle completion, we encounter the microbial architects who specialize in transformation, the microscopic artists who create beauty through change, who build new possibilities from old materials, and who demonstrate that destruction and creation are merely different aspects of the same fundamental process of renewal and regeneration. Throughout the natural world, microbial architects serve as the engineers of metamorphosis, constructing the biochemical pathways that allow dramatic transformations to occur. These metamorphosis engineers work behind the scenes of some of nature's most spectacular changes, providing the molecular machinery that makes transformation possible. In the soil beneath decomposing leaves, metamorphosis engineers construct elaborate biochemical networks that transform dead organic matter into rich humus that can support new plant growth. This transformation process involves the coordinated work of hundreds of different microbial species, each contributing specialized enzymes and metabolic capabilities to the collective project of renewal. The transformation of organic matter by microbial metamorphosis engineers is not simply decay, but a creative process that builds new forms of complexity from the raw materials of death. Complex molecules are carefully disassembled and their components reorganized to new structures that have different properties and functions than the original materials. Marine metamorphosis engineers construct the biochemical pathways that allow dramatic changes in ocean chemistry and ecosystem structure. They can transform oxygen-rich surface waters into unoxic deep-sea environments, Create the conditions for coral reef bleaching and recovery. Facilitate the seasonal transformations that characterize marine ecosystems. The work of metamorphosis. Engineers demonstrates that change is not random destruction, but organized reconstruction guided by principles of efficiency, sustainability, and renewal. Their transformation projects create new possibilities while preserving essential elements that can be incorporated into future construction activities. When environmental conditions change rapidly, specialized microbial architects work as adaptation facilitators. It's constructing the genetic and biochemical machinery that allows organisms to develop new capabilities and adjust to altered conditions. These facilitators demonstrate that adaptation is not a passive response to environmental pressure, but an active construction process guided by molecular architects, mutation facilitators. Construct specialized DNA replication systems that can increase mutation rates when environmental stress indicates that rapid genetic change might be beneficial. These systems allow controlled increases in genetic variation while maintaining the ability to return to normal. High-fidelity replication when stable conditions resume. Horizontal gene transfer facilitators construct molecular machinery that allows the rapid exchange of genetic material between different organisms. These systems enable microbial communities to share adaptive innovations and respond collectively to environmental challenges that might overwhelm individual species. Epigenetic modification. Facilitators construct regulatory systems that can rapidly alter gene expression patterns without changing underlying DNA sequences. These systems provide flexible responses to environmental changes that can be rapidly implemented and easily reversed if conditions change again. The coordination of adaptation facilitation involves sophisticated environmental sensing systems that can detect the types of environmental changes that require adaptive responses. Adaptation facilitators must distinguish between temporary fluctuations that require only short-term responses and sustained changes that justify the costs and risks of genetic modification. At the deepest level, microbial architects work as renewal specialists, constructing the fundamental processes that allow life to continue indefinitely despite the constant challenges of entropy, environmental change, and resource limitations. These renewal specialists demonstrate that life is not a temporary exception to physical laws, but a sustainable construction project that can continue indefinitely through careful management of energy and materials. Cellular renewal specialists construct the molecular machinery for DNA replication, ensuring that genetic information can be accurately copied and transmitted to future generations. Their construction work involves proofreading mechanisms, repair systems, quality control processes that maintain the integrity of genetic blueprints across countless generations. Metabolic renewal specialists construct the biochemical pathways that allow cells to continuously rebuild their molecular components using energy and materials obtained from the environment. Their work demonstrates that living systems are not static structures, but dynamic construction projects where every component is continuously being replaced and renewed. Ecosystem renewal specialists construct the nutrient cycling systems that allow ecosystems to function indefinitely without depleting their resource base. Their work creates closed-loop systems where waste products become raw materials for new construction projects. It's demonstrating principles of sustainability that human technology is still learning to emulate. Evolutionary, renewal specialists construct the genetic and ecological mechanisms that allow life to continuously adapt and diversify in response to changing conditions. Their work ensures that life remains flexible and creative, capable of developing new solutions to novel challenges and exploring new possibilities as opportunities arise. And so, dear listener, as our long journey through the realm of unseen architects draws to its gentle close, we find ourselves returning to where we began, to the quiet recognition that we are never alone, never separate from the vast community of microscopic builders who surround us, support us, and sustain us in every moment of our existence. As you settle deeper into rest, let yourself feel the presence of these invisible companions. In the soil beneath you, billions of bacterial and fungal architects continue their patient work of building the foundation that supports all terrestrial life. In the air around you, atmospheric architects process gases and regulate the composition of every breath you take. Then your own body, countless microbial partners work through the night to maintain your health, support your immune system, and even influence your dreams. We have traveled together through microscopic cities more complex than human metropolises, witnessed construction projects that span geological ages, and observed engineering feats that surpass our greatest technological achievements. We have seen how these unseen architects build landscapes and regulate climate, facilitate evolution and maintain the conditions necessary for life, create beauty from decay and construct hope from destruction. The invisible architects work in harmony, not competition. They demonstrate that the greatest constructions emerge from cooperation, that sustainability comes from circular systems where waste becomes resource, that resilience emerges from diversity and redundancy, and that beauty can be found in the most microscopic details of life's endless creativity. They teach us patience, their construction projects unfold over time scales that make human endeavors seem brief moments. They teach us humility. Individual microbes are tiny and seemingly insignificant, yet their collective efforts have transformed an entire planet. They teach us interconnectedness. No organism works in isolation. All contribute to the success of the whole community, and individual well-being is inseparable from collective flourishing. As you drift into sleep, carry with you the wonder of this invisible realm. Remember that you are not merely observing these microscopic architects from the outside, but are yourself a walking ecosystem, a mobile biosphere that hosts hundreds of species of microbial partners. Your existence is a collaborative construction project, a partnership between human cells and microbial architects that creates something more beautiful and complex than either could achieve alone. In your dreams tonight, perhaps you will walk again among these tiny builders, witnessing their patient work, marveling at their ingenuity, and feeling deep gratitude for their endless contributions to the world we all share. Perhaps you will carry into your waking life a deeper appreciation for the unseen architects who make every moment of existence possible. Every breath of air breathable, every grain of soil fertile, every drop of water clean and life-giving. The construction work continues while you sleep, as it has for billions of years, and as it will for billions more to come. The unseen architects never rest from their work of building, maintaining, and renewing the world. They are the true immortals, the eternal builders, the invisible foundation upon which all visible life is constructed. Rest now in the knowledge that you are held and supported by this vast community of microscopic architects. Rest in the understanding that every cell in your body carries the wisdom of billions of years of construction experience. Rest in the peace that comes from knowing that you are part of something far greater and more beautiful than any individual existence, part of the grand, ongoing construction project that is life itself. Sweet dreams, dear listener. May your rest be peaceful, your sleep be deep, and may you wake tomorrow with renewed wonder at the invisible architects who shape our world. In every grain of soil, in every drop of ocean water, in every breath we take, invisible architects work tirelessly, building and rebuilding the world we know. And in our sleep, we join them in their endless construction of tomorrow. Thank you for joining us on this contemplative journey through the realm of unseen architects. If this gentle exploration has brought you peace and wonder, please consider subscribing for more sleep-focused educational content. Until we meet again in the microscopic realm, rest well and dream of the invisible builders who never cease their work of creating and maintaining our beautiful living world.